say for the introduction. Hello everyone. Thank you all for coming. We are sorry we don't get a bigger venue for this, but hope you're all comfortable on this Tuesday night. Alright, today we have Professor Alain from Oxford Math Institute, and he'll be talking about neurodegenerative diseases and models about that. Yep, as well as brain dynamics and other interesting stuff. So if you can, please give a round of applause to Professor Alain.
Wow, it's not every day you perform in front of standing room. That's great. Thank you very much to Kai and to the Oxford AI Society. I'm not going to talk about AI, so I hope you won't be too disappointed. I think that maybe the important part about doing good AI is to understand OI first. So I have some idea about AI, but today it would be organic intelligence, not artificial one. And I'm interested in a series of problems. Today I'll be talking about dementia, but I see dementia as a way, as a window, research in order to understand cognitive function. So it's joint work. It's funded by the EPSRC mostly, the Research Council, and it's collaborative work with a number of people around the world, primarily in Stanford and in Germany. So to start, this is a very nice, it's an art piece, but it's based on medical imaging. This is a sagittal slice of the brain. So just to introduce the topic, what you see right away is these long filaments that radiate from the outside. So the outside of the brain is called the cortex. This is where you have a large number of cell bodies. And these cell bodies have these long extensions, which are called axons, that essentially connect part of the brain to itself, to other part of the brain, or part of the brain to the rest of the body. Like, for instance, the very large axon that you have here on the top of your parietal lobe goes down to the motor cortex, goes down all the way down to your legs, as you see coming down. So this is the overall global structure. We're going to try to understand it a little bit more, how it works and what we can say, but also how it can be affected by disease and what happens during that time. So it's part of a broader research program that I've initiated about 4 or 5 years ago with a number of collaborators around the world. And the general philosophy is to try to understand various aspects of the brain that people have not properly addressed in the past. There are very good models at various levels in mathematical neuroscience of the brain, viewed as transfer of information. But we thought that there are aspects related to the physics, the chemistry, the mechanics of the brain that have to be integrated to have a better understanding. So our approach is to look at the brain at different levels and see how these different levels have implication on each other, going all the way down to the molecule, the protein side, to the cell level, to the tissue level, to the organ level, and the organism. Remembering that the primary function of the brain is cognitive, we want to see how an event that takes place at this level impacts our cognitive function. And what we talk about today is going to be an example of that. So our motto, in a way, is that really understanding some aspect of the brain, maybe not all aspects, require multidisciplinary approaches, obviously, but also multi-scale, that you have to link these different scales, which is particularly important. A lot of process in physics take advantage of the fact that you have a decoupling of scale. The law of gravitation, the motion of the planet around the sun, do not depend on what happened on the sands on the beach. You can forget about, you can think of that as one scale, right? But most of the phenomena in biology require understanding at a very, very small scale. So understanding what happens at 10 to the minus 6 or 10 to the minus 10 meters as an application at 10 to the at the level of the full organism. No, we have a party. You can actually lower it. Can you just lower it a little? Because this is more important than me. So I just want to make sure you see. Yeah, that's good. Multi-scale methods, but also multi-physics, that there are a lot of different physical fields that have to be integrated in order to get a better picture of that. And there was really a hole, despite the huge amount of funding and research going on in the brain, with that kind of approach. And in order to fill it, we just started, actually it starts next week, a new journal called Brain Multiphysics. And it's myself together with Antoine Jerusalem within the engineering department. A few years ago, we started a kind of network for people interested in this topic. And now we have a journal for that community. So, but today's talk, I will first talk about dementia. I will talk about mathematical models of how can we approach the problem. I will show once we have developed these models, so we can simulate it to have the real evolution of what happens in the brain. We can further simplify this model and use aspect of the network theory, really trying to extract the main feature of the process, and then from that trying to understand the effect of this disease on cognitive decline. Okay, so in 5 easy steps. So let's talk about dementia. Let's go back to what is really patient 0. So this is August Datton. In 1890, she started showing very troubling signs. She couldn't remember basic things, names, places. She would wake up in the middle of the night, screaming, dragging sheets around the house. She would have period of apathy. Eventually, she was admitted in a mental asylum called the Herrenschlosses, the Castle of the Insane, really, close to Munich. And we have very good record of interaction or of interviews with doctors and all that, in which one of the things she said, thinking about what happened to her in a moment of lucidity, she says, I've lost myself. It was really typical of this dementia. This is early onset dementia. She was only in her 40s. That the patient really lose part of themselves. It's like evaporating. The personality, the being disappears. She died in 1910, and the doctor that took care of her wasn't there anymore, but got the sample of her brain. And thanks to the progress of medical imaging at the time, which means proper staining, so to see different type of protein, a doctor, Alois Alzheimer, managed to isolate and for the first time diagnose the disease that she had. And he called that not Alzheimer's disease, that would come a little bit later. He called that the disease of forgetfulness, because Alzheimer's disease, the primary symptoms, so the primary cognitive effect, cognitive decline, is always related to amnestic syndrome, or loss of memory. But the diagnosis, and up to 2007 was the main diagnosis for the disease, was that he found the presence of 2 types of protein, aggregates of protein within the tissue. So there was something physical, it's not just aging or fast aging, there was something that was disease within the brain. And what he found is the famous amyloid beta, which has aggregate of extracellular protein, and tau inclusion, which has intracellular aggregate of protein. They sometimes called neurofibrillary tangle. So these are very large bundle or aggregates of proteins. So it's important to reflect also on the effects going from T's inclusion on the personality. And a good example of that is William Mullen. He was a painter, was an American painter who was born in Philadelphia in 1933. And he immigrated to Oxford. He came to Oxford to the Ruskin School of Art and then had a very successful career as a painter with the use of very vivid imagery, pop and very vivid colors. But in his 60s, so the early 90s, he started showing a few signs that were worrying. He couldn't properly tie his necktie, he couldn't do his house finance, he started forgetting things, So he went to see his doctor, and the doctor said maybe it's depression, maybe it's other thing, and ordered a series of tests, including MRI, including some blood drawn and cerebrospinal fluid. At that time, with the different evidence, he was diagnosed as having Alzheimer's disease. But he decided with his wife, who was an art critic, to document the progression of his disease through his own painting, through his own self-portrait. And this is the progression. So 96, it was still, you can see the use of color in fully able to depict himself. It's very nice self-portrait. This is 97. This is 98. You can already the fast progression of the disease. 99, and the last painting is 2000. Right? The Alzheimer's disease starts in the temporal lobe, in the entorhinal region, and quickly affect the temporal and parietal lobe. The interface of the two is where the spatial visual representation of the world is. So a patient loses ability to fully represent the world around him. And you see that both the personality, the different symptoms appearing, the way he reflects himself, the way he's able to handle his art, his tools, and the way he can see the world. Patients typically have a hard time representing three-dimensional objects. Other types of neurodegenerative disease do not have that, and sometimes, for example, frontotemporal dementia, patients suddenly exhibit actually very good talent for a short period of time. But this is very typical. It's a good representation of what happens to the patient. It's in a way the ultimate representation of the self. And it's a good way to talk or understand staging in the disease. So staging is the progression of the disease. So the typical cognitive staging is that first you have, in the first stage, amnestic syndromes, so problem with memory and recalling memory. So it's not like, oh, I don't remember the name of that person I just met. It's a memory where even when you are cued, or you are given cue or hint about something, you were unable to recall it. So one of the tests is you show animals, for instance, and you have to say this is a coat, this is a dog, or something like that. A patient with Alzheimer's disease might lose not only the recall of the name, so short-term memory, or just naming, but really semantic memories, meaning the meaning of the object. If you tell where, we make beef, it goes milk, it goes moo moo, or that, still no idea. What is, it's an animal, no idea, no cue, no idea. So the meaning of the object disappears. So that's the first stage, and now it's one of the main tools in order to pre-diagnose Alzheimer's disease is to go to this test with recueing. Since 2007, it has evolved. The second stage is a cognitive problem related to problem solving, abstraction, things like that, relating to the world, trying to make sense of things. After that, you have more problem directly related to what's called instrumental problem. You have aphasia, agnosia, apraxia, and so on, inability to recognize face, we have object, to recognize words, inability for fine motor control, and in the last stage of the disease you have large psychobehavioral problems, long period of vegetative state, apathy, behavioral trouble, burst, violent outburst, and so on. And these stages are reflected by actual physical change in the brain. Early on, it starts, so T is a neurofibrillary tangle of the tau protein I told you about. The black one, the black is a stain for that. So this is where it starts, this is the hippocampi. So this is where a lot of memory is stored, and the first effect will be seen there. The next stage, it starts evolving to other area of the cortex, and eventually it infects all the cortical area. And now we understand well this stage, Brack and Brack, wife and husband, have through 20 years of accumulation of data of pathology, or slice of diseased brain, have mapped the progression of the disease. What's fascinating about neurodegenerative disease, one of the first things that puzzled me in a way, is that T's are very systematic. 85% of Alzheimer dementia are systematic progress to the same stages within the brain. So it's called a homogeneous disease. It goes to systematic phase in the brain, and they've been catalogued. So in T's, that was a coronal slice. If we go back to a sagittal slice of the brain, you have the entorhinal cortex invaded early on, hippocampi, goes into the frontal lobe and starts invading where you don't see the parietal and temporal lobe, and then eventually all part of the brain are infected by the disease. And you can see here, for instance, that the parietal lobe, the one that controls the motor control, is one of the last affected by the disease. That when the patient can still walk, where there are other neurodegenerative disease, this part of the brain will be affected first. So it's important to look at that in a global setup. Alzheimer is by far the most common. At least 60% of all cases of dementia are thought to have a component of Alzheimer's disease. But there are other neurodegenerative diseases where you have the same kind of systematic infection, systematic invasion of toxic protein within the brain. In the case of Parkinson, it's the alpha-synuclein. It's another small protein, intracellular, that goes to the brain stem and start invading the basal ganglia, the amygdala, and all the process related to motor control. That's what you have early tremor and things like that, and eventually invading all the brain. By the end of this disease, they all start looking the same because all the cortical area have been invaded. ALS, Lou Gehrig's disease, there it's much more directly related to the parietal lobe, so you see the effect right away in terms of motor control. It's associated with another protein, also TDP43, and that again you have an invasion of the brain overall. So T's are not infection. This is not something you can get from somebody else or from eating bad meat or anything like that. But they're somewhat related to prion disease. And prion disease are protein disease that are infectious. So this is Matco disease, for instance, Creutzfeldt-Jakob disease, is one that you can get from eating meat or brain. Kuru is the laughing disease. It's associated with cannibalism in the Polynesia, and so the disease would be carried by when someone would eat somebody else's brain who had the disease. It's called the laughing disease because it affects one of the places that it affects is the self-control of laughter, scrapping sheet, and so on. So these are all different types of protein, and they attack different parts of the brain. So it's good to keep that in mind. I'm also, as I told you, interested in the physical aspect of the brain, what happened, not just a collection of cognitive or histology. What really happened in terms of the development of physical force and the change in the brain. So what we know about normal brain, so this is a data set from 1978 out of 2,000 autopsy reports for each man and woman, men in blue and men and woman in red, that shows the average brain weight as a function of age. So what you see is, at first, very early on, you got a very rapid increase in brain weight up to the age of 20. Okay, so if you're less than 20, congratulations, the brain is still growing. And that's followed by a small, a very progressive loss of brain weight all the way down to the 80s. And the total mass is not that much, actually. It's only 11 or 15% for men and women. And it's mostly not neuron, you lose only 2 to 3% of neuron. So normal aging is perfectly fine. You can be very healthy, like this guy. Because if you stay a little bit too long on a TV show, you end up like this. You have to be careful. That's a little bit more than 11%. So normal aging, the brain is mostly fine. What you lose typically are maybe glial cells, so that explains why old people might be a little slower, because there is not as much control of the neuron for transmission, but there is nothing wrong with them. So respect to elders. But this is the difference with Alzheimer's disease. On the left is a healthy brain, well, it was healthy up to the moment it died, so it didn't die. By that, a non-brain of a dead person who didn't die from brain disease. And here's the Alzheimer. You see right away the big difference, right? Completely shrunk. There is massive atrophy that takes place in this person. And you can see that better on these, if you look at a coronal slice and compare one next to another one, you'll see right away a number of features. You have larger ventricles. So ventricle is where the cerebrospinal fluid is being created and recirculated, so that to maintain an intracranial pressure, it bathes the entire brain and maintain proper function. You have an opening of the sulci. Is that this feature, this value is in the side. So here the gyri at the top and the sulci at the bottom, and really here you see them opening. You have a thinning of the cortex, and that's really bad because most of how your cognitive function takes place within the cortical area, and the same is true for the hippocampi, that will take place very early when memory is stored. So this happens in your brain, right? It's a progression of the disease, and no amounts of eating blueberry or broccoli is going to reverse that. So don't believe everything you read on the internet. Another important feature is to obtain global way of describing the evolution of the disease. These are called biomarker. And biomarker is obtained either to blood or cerebrospinal fluid or to MRI. And so this is a function of time. And if you can measure that, you reach a certain point where you reach a certain level of detection threshold. And you see that different biomarkers will exhibit different type of progression. Some are expressed more earlier on. So if you are able to monitor amyloid, beta amyloid, this will be the first thing to show that there is a problem with somebody, way before you start seeing cognitive problem in green. In reality, what happens is that people show cognitive problem, and then they test it for the other one, and they show very high level, because people are not tested. People don't go every year to get an MRI, or CSF is not obvious. To drain CSF is not particularly pleasant or anything. It's not just like blood work. So, but Dr. Jack came up with this curve explaining that essentially what you would expect for the biomarker is they start low and increase and go up. I said, that's great, the problem is solved. We have a model that explains this, except this is a model view from clinician. That means you go look at the paper reference, and then you realize that the idea of doing a model is that they draw on a sheet of paper on the computer this curve and say, that's what happens. It's fine if you're a doctor, that's a way to express an idea or a model, but I can tell you as a mathematician, you're not allowed to do that. You're not allowed to say, this is what happens, and draw a function on the screen and be done with it. So what is that based on in terms of data? You can say, well, maybe there are plenty of data and that's just a fit. This is the best set of data that we have. So you see that's the PET, that's CSF and all that. I don't know if it's just me, but I don't really see this curve just jumping out right away for me. And this is a very nice set of data, because here is the years of onset. So the first time the patient was diagnosed was at this point. And you can ask, how did they know what the curves were before? Well, these are people who have a genetic variant of the disease. It's quite rare, but they exhibit very early on the disease. And so since there's a genetic marker for it, some of these, there are only 250 families in the world who have them, they can be followed way before the disease, about 30 years before. There are certain type of variant, genetic variant, that for which the symptoms appear exactly at age 49. Age 49, your parents or your father, your mother starting having dementia at age 49, and you have that genetic variant, you have the same problem at age 49. So that's what but it's still pretty far from what you expect. So for me, when I looked at it, I say, well, the best I could say is that I have a line like that, maybe a line like that or like that. I have no idea, right? For me, it is a very different type of behavior, and I want to find them. So in terms of mathematical modeling, what the challenge is, I want to understand the staging. I want to understand how the disease progresses within the brain. Is there a general way to approach that and understand that for different disease? I want to understand from the staging if I can predict atrophy within the brain, and I want to understand the evolution of the biomarker. And that would give me a general picture of the physical evolution. And based on that, I want to go one step further. I want to be able to see what I can say about the overall cognitive decline of the brain. And we'll talk about that a little bit. Okay, so let's try to do some very basic mathematical modeling here. So an idea that came from one of our collaborators was one of the early papers of 2013, 2015 for this type of disease. It's called the Firelight Hypothesis for neurodegenerative disease. And the idea is that it's these toxic proteins that are responsible for the disease, just like in the case of prion disease. And that these toxic proteins mostly diffuse along the axonal pathways, this long filament that we saw in the first line. And what happens is that just like prion, when they meet a healthy protein, they can template and convert them to a toxic protein. That's what we know from prion disease. So, for instance, the tau protein that I show you, that I talk about, it is a normal protein that the brain uses for normal function. They stabilize the microtubule within the axon, and they're naturally produced by the cells and regenerated, clear, and all that. But if you have a toxic one, the idea, the hypothesis, is that a toxic one, when it meets a healthy one, converts the healthy one into a toxic one, just like in a bad zombie movie. Zombie is the bad guy, meet a normal guy, bite them or whatever, and they're both zombies. And then you have two zombies, they meet more people, and you know what happened in these movies, right? You have an autocatalytic process, and soon everything is converted. I don't know, they never end well. And this never ends well either. Essentially, there is no cure for Alzheimer, for instance. The last week there was some news that there may be some hope about some treatment. There are 4 drugs, but it's mostly to try to treat some of the symptoms. So there is essentially no cure and very little treatment. The disease progresses within 5 to 10 years. Typically, it starts at the age of 60 and run its course until the person dies. So the idea is that when a toxic protein invade the tissue, they reach a certain critical mass where they damage the tissue, the normal function of the tissue. The cell die, they remove from the brain, and atrophy takes place. So the idea of the model is just to follow the basic description from this early paper on transport and creation of protein, and then couple that, a transport model, to an atrophy model. So again, the basic kinetic model, one of the simplest ones, we have different ones with different type of resolution, is that you have a healthy protein in blue, and then you have a misfolded one in red. They meet, and the red one converts the blue one into 2 red ones. And then they fragment, and now you have 2 red ones, and they start doing the same. So if you think about that, it's one big step. I call P the concentration of healthy, P tilde the concentration of misfolded. I have P2 plus P tilde is converted in one step to 2P tilde, P tilde plus P tilde. Right? So it's a very simple chemical process to model. So the way you transform a chemical equation into a mathematical equation is that you look at the rates of concentration, and you say that the rate of concentration has a part that's normal production, so that's normal tau being produced by the brain. It has a normal clearance rate, they produce and they remove at all time, so that's normal business. If it was just that, that would be healthy. But that when it meets, when P meets P tilde, it's converted. So you remove that, the rate of change is negative, that's what is minus sign, at a rate K12. The toxic protein is created, is also clear, but it's only created, there is no source term by itself, it's only created by by the healthy protein. So you have a clearance term and you have a conversion term. Okay, so let's start with this model. I will talk in a second about what these terms are. These are just for transport. It's a diffusion term. But if P is much bigger than P tilde, you can simplify that. And now you have a population C, which is the toxic protein concentration. And you can relate the parameters, it's not important. No, C is the concentration of toxic protein. Now it's rescaled, it's rewritten in a mathematical way so that C goes from 0 to 1. 0, no toxic protein, 1, the maximum amount. So it has a production term and then a negative term minus C squared. If you look, it has only one parameter, which is very good because it's very hard to validate any of these. So if you've done any mathematical biology, you certainly have come across an equation like that or very similar. This is a version of the Fisher equation dating back from 1937. Fisher is the great one of the great British statisticians. He came up with this equation to explain the evolution of genes within a population. But the same equation it's like the Newton equation of mathematical biology. It's used everywhere. Population invasion, disease progression within a population. This is the kind of equation or idea that is used nowadays in the big center for disease control, etc., to understand the evolution of disease on the world. Right? This is really the basic equation for that. There is a little twist. It's anisotropic because the progression is much faster along the axon, and they're very nice experiments to do that. So remember, you have the cell body in the cortex, and they connect with this long filament axon to other cells or to other processes. And you can do experiment where in blue you add this tau protein with a marker, with a GFP, and then you can see them going very quickly. You have very fast axonal transport along the axon and very slow off the axon because they leak a little. So T's operated diffusion is not simple diffusion like heat in this room, which is isotropic. It's just like a way it says, well, if I had a bunch of conductors, the heat would go very quickly in this direction and very slowly in the other direction. Right? So imagine that here I have a bunch of rods, metallic rods, and the conduction would be much faster in this direction than around the other one. And this is an important feature because it really relates to how these proteins are transported. Forget about the equation. The equation are not important. It's the concept that they carry through. What we know about this equation, we know an awful lot. I mean, it's a major topic of research, both in mathematics, in mathematical biology, in biology, and so on. What we know is that in one dimension, just along an axon, what would happen is that if you start with any small concentration, look at the red patch, anywhere, any small amount of toxic protein, so that little hump is just the amount of protein, this is what you would obtain. In time, it would evolve, go to maximum value, C equals 1, and start filling on both sides. That's the solution of the equation. And it's very nice because you can relate the velocity of propagation to the parameter D is the diffusion along the axon, and alpha is the one parameter that explains how quickly the population expands. So that's good. I show you different pieces of a model. And so again, it's just a mathematical way. Again, the mathematics of the equation is not important. But what it says is very simple. It says that toxic protein, if you start at some point, will be transported very quickly along the axon. And whenever they meet healthy protein, would convert them into toxic protein. But that's okay. But now what we need is that out of that, what we need is to move that up to the entire brain. What happened at the brain level, right? We want to upscale. So we need a brain. There are plenty of brains around, on which to run this equation, essentially. And that can be done. You place somebody in the scanner, that's Ellen, when we were in Stanford, And what you do is you go zoop, 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 zoop. It creates image, black and white picture. And out of that, the second step is to segment it, is to transform this black and white pixel image into mathematical domain, little triangle in 2D or tetrahedra in 3D, doesn't matter. This is a very nice problem for AI. It's not completely solved because there are a lot of subtleties, but it's a typical problem of machine learning to recognize different levels. And there is a lot of work in this area. For instance, MRI, how to recognize where there is a tumor, for instance, things like that. This is the typical kind of success that machine learning has. So you have all these 2D slides. You can combine them in a 3D picture. This is the white matter of Alain's brain and the cerebrum in green. And so you can add different layers. So this is the gray matter, and this we also have the skull. And we use this one to do a simulation of trauma and craniectomy and swelling and things like that, also for other applications. But you also have more information. You have information about axonal direction. There is a type of medical imaging called diffusion tensor imaging that allows you to find the direction of fastest transport of molecule within the brain. And what we know is that it's the fastest along the axon. And so what you obtain is this type of information, where at each point you have a little vector that tells you that direction of fast transport. And then you can piece them together in order to try to find axon and axonal bundle. So you have that information. Now, without cheating, this is what we can do. We can take the same model and make the hypothesis that the only difference between different neurodegenerative disease is not the process itself, it's mostly where the disease starts. And we can start with the chart of the disease that people have known, we're trying to see where it starts, and just let it run without changing any parameter for a given disease and see how it compares to the invasion map that are known from clinical histology. So depending of the disease and the type of protein, you have different type of starting point. Tau inclusion starts around here, TDP in the parietal lobe, alpha-synuclein for Parkinson in the brain stem, and so on. And amyloid beta very diffuse. So this is, for instance, a 3D simulation of the evolution of the disease. If you run it using finite elements, and you see that red is bad, of course, blue is very, very slow. This is at mid-stage when you see the whole profile, and that's in time when you just see the, let's go back and see it again, because it's kind of cool. Also took 2 weeks to make, so. So I want to get some mileage out of it. Okay, so now we can do that. And we can go back to the Firelight Hypothesis and all these maps and look at the evolution. So the only thing that we start is the initial seed and we let it evolve. And great thing that you see right away is that the colors really match. So that's a very good start. So don't let it fool you. But what you start seeing is that without any assumption, with a model that's admittedly very crude at the modeling level, we're not saying really anything special, it captures all the main features that are seen with this disease. The arrows that you see in invasion really reproduce what you see. Here, we had disagreement to what happened in the occipital lobe for ALS. I don't know why. But I'm discussing with experts on that. That picture might not be as accurate as people believe either. So we don't want to cheat. This is what it is. I'm not going to mess with or change the parameter in this region so that it's why that wouldn't be honest. The main point is that you really capture everything, but you also recover T-curve, which are the, now you know all the parameters because you have a full simulation. You can average that over the entire brain. You can look at the evolution of amyloid beta, of TDP43, of tau, and so on, and you recover essentially this Jack's curve. So Jack was quite... I'm not saying he didn't have a good intuition, he was very smart. What I'm saying is that this is a way to recover in a consistent way based on simple assumption at the local level. So that's good. Now what we want also is to recover atrophy. What can we say about atrophy? So what we do is we start with an initial seeding. I'm not showing the seeding here. What I'm showing you is it's another simulation of the change of the brain as a mechanical object. We seed it and we let it evolve, and we couple the concentration to an atrophy model where we remove part of the volume element. And this is what you have. So the red shows high level of stress, not high level of concentration. But you recover all the features: opening of the sulci, thinning of the cortex and the hippocampi, enlargement of the ventricle. And you can do that in 3D. So that was good. We recover all what we said to do. But then the question is, great, my model works. Why does it work? What is the essential feature that are important to make it work? Can we further simplify it so that we really understand what's in the brain makes it so special? And the assumption that we had to do for this problem and the other one is that what's really important is the intrinsic geometry and topology of connection in the brain. That's how things quickly go from one place to the other one. And so the next idea was to try to simplify further the model using the full model as a test, and to look at the brain not as a continuum but as a network. And the idea, going back to the DTI, this mesh, is to look at different regions of the brain. And you can do that with brain atlas. You see here is the frontal pole, here is the entorhinal region, here is the occipital, here is the gyrus. So you can, there are different resolution of that. And then you look at all the axons connecting one region to another. So you look at this connection. And then you forget about the green picture, and what you're left with is just nodes, so the region with the connection. And you count how connected they are. You count the length, and you count the number of axons going from place A to place B. And so that gives you now a graph, a full graph, but that's just a picture. You want to go a little bit further, so you give names to each node, 1, 2, 3, 4, 5, 6, and for instance assume that it has this connectivity. What you want is to transform that in a mathematical object on which you can operate. So what you create is called the adjacency matrix, a connection matrix, and the rule is very simple, so you can always do it together. So let's look at 1 and 2, for instance. 1 is connected to 2. So if I look at 1 and 2, I'm going to, in that table of number, I'm going to put a 1 in first column, and first line, second column, and second line, first column. So it means 1 is connected to 2 and 2 is connected to 1. I'm not doing directed graph here. So every time 1 is connected to the other, so it's a symmetric matrix, as you see. Right? We all agree on the rule. So for instance, 6 is only connected to 4. So what you expect to see is that line 6, column 4, you have a 1. Yes, good. Line 4, column 6, you also have a 1. Sorry, let's go back to it. We'll go back to that in a second. 1, 2, 3, 4, 5, 6. Up, graph, black. And 1. Okay? So we can we all, you agree that we can, with red pencil and a graph like that, you can start playing and creating this matrix. This matrix contains what we call the topology of the graph, the essence of the connection. It's just a table of number of 1 and 0. It really reduces the brain to its essence. This is an 83 nodes, so you have an 83 by 83 matrix of 1 and 0 that somehow encodes something about how the brain is connected. And when you think about all the complexity of brain, neuroscience, neurosurgery, biochemistry and all that, this is the most essential reduction of a representation of the brain. There is nothing simpler you can do about the brain than that. There's a little bit more complicated. Here is 1,000 nodes. So this is average over 429 healthy patients that you can download from a publicly available database. And that's the weight matrix for that in color, because colors is prettier. And it also contains some information. So blue is 0 and red is 1. And in between, it tells you it's something related to the number of fiber between two regions and also the length. So it has a little bit more information. It's a weighted one. And what you see right away is structure of the brain emerging. right? When you see this matrix, you can already say something about the brain just by looking at it as a picture, which I think is amazing. Whenever I see that, I say, wow, that's amazing. Look, we see a red and blue matrix, and we can say something about the brain. What can we say? Where RH is for the right hemisphere, LH is the left hemisphere. And what you see right away is that the two are poorly connected. It's almost entirely blue here and blue there. The points that are not blue are this part of the brain, the corpus callosum, that connect the two. That's where the connection occurs, right? There's much more structure when you start looking about network. It has a small kind of 6 degrees of separation, the two people in the world. This idea of small world network and things like that had become popular about 20 years ago when my friend Stephen Strogan started playing with that with his student Duncan Watts. Well, you see the same structure in the brain. Very tightly connected to its neighbors. That's why the red line is very red around here. So that says neighbors are highly connected. And then with a few connections to other places of the brain. You have the same type of structure. Yes?
How much does this vary between individual brains?
So that's good. That's a very good question. So that's why we do an average. If you take different patients, on average, the number of connections that you will find is 3,000 for each of these 400 brain. And this one is 70,000. So one brain to another. The question is, do you not have the connection because you don't have the medical imaging is not good enough? Or is it because you have individual variation between people? It's not quite clear. But we're looking at that, and we're applying some idea from topological data analysis in order to try to see, as you reduce the weight, we can say something about the variability of these matrices within a population of matrices. It's a very good question. You should feel free to stop me and ask me a question in case you haven't noticed. Thank you for showing the way.
Maybe I missed it, but how do you decide how many neurons or nodes you need? Because those are probably not the number of neurons you have in your brain.
Oh, no. So we have 86 billion neurons about. So these are regions that are evaluated, so typical region that people have been studying for more than 100 years, that have been isolated as being important for certain region. So related to language, related to the process and all that. So these are entire region, and the connection are not individual neurons, but big bundle of neurons containing thousands. So it's very different approach than the Human Brain Project, for instance, that try to simulate little region of the brain where you take care of all the possible connection, 100,000 or something like that. This is really stepping out and saying, okay, I'm not that interested in the detail of this connection, but the overall connection between different regions. If each of these balls can be themselves analyzed or modeled as interaction of a lot of different processes taking place. But MRI wouldn't give you that. Right? It's very crude for EEG. Okay, so it's still turning, that's good. But we want to do a little bit more. Remember, what we want is to be able to represent transport process of diffusion along. Again, the idea of the Firelight Hypothesis is that a protein would go very quickly to this axon. So if I give you a graph like that and I tell you the length, the idea is to see how something would be transported. Could be heat, could be concentration, or anything. So I would like to know, if I start heating my system, what would be the distribution of heat at a later time? Pure diffusion process, for instance. Where there is a natural way to do that, that's well known from graph theory or from network theory, is that from that matrix you can create another matrix. So from W you can go from L, and the construction is very easy. Whenever I have 1 here, I put -1. Okay, we can do that. And then I count the number of 1, the number of 1 there. There are 2 1 on the first line, so I put 2 on the diagonal. There are 3 1s on the second line, so I put a 3, and so on. Okay, can all do that. And what's special about this matrix, it has a name, so we know it's special, is the graph Laplacian. This is the discretization of the diffusion operator on a graph. It's much more than that. The graph Laplacian is central in all graph theory. It contains a lot of information about what happens on a graph, how information is being processed and all that. So this is the natural object for the propagation of transport for us, of the physical transport. Now the reaction part is the same, and I can now have a simple system of equations that describe the evolution. So I have a way to diffuse, I have a way to integrate it. Now the system is much simpler. Before that it was partial differential equation, very hard, big computer, big software, lots of problems. This has no equation that are on the nodes, so they are ordinary differential equation, much simpler. If you can program your iWatch, you can run on 83 nodes on that, and let it run. And so you can really push and study that in more detail and have nice mathematical results. So here is our BrainNet 1.0, which does the propagation and autocatalytic reaction on any kind of network. So this is 1,000 nodes. And what we do here is that we seed in the same region that the continuous problem for tau protein. And when we seed for tau protein in the entorhinal region here, and we let it evolve in time. So we see both left and right because there is no known asymmetry in the disease. So the one on the right is the edges, what happened on the edge, and the one on the left is what happened on the node, and the left and right hemisphere symmetric. And then you have invasion of in the entire brain. So that's a very nice tool because now you have the clinical data with this evolution, you have the continuum model that we talk about, and you have a discrete model where you can look at the evolution of this protein. Now you can go back and look in more detail on the evolution of the graph, on the network, and you recover essentially all the aspects that I told you. But you can, in a very nice way, you can do some mathematical analysis and show that the primary infection in the brain at first is only connected to nodes that are directly connected to each other, so first neighbor. And it's a purely topological property that's independent of the detail of the kinetic. And then you start realizing why the previous models work. They were capturing the essential of the topology. What this analysis shows you is that whatever the kinetic is not that important. As long as it has some generic feature, what is important is the connection to explain how the invasion first takes place. It goes from limbic to basal ganglia to temporal lobe. And then you have a secondary infection that you can show really depends on the details of the kinetic and the transport process. So that part is related to geometry, notion of length, and chemistry, whereas before it's mostly topology, when all the two things are connected, know how far they are. And that's where you have further invasion, parietal, frontal, and occipital lobe. So you recover all these aspects. But you also, what you naturally generate is an evolution of a network. The network, you can think of the network also changing in time. And that's very interesting because now you can think about cognitive decline. So that's preliminary results over the last couple of months or so. But here is the basic idea. This is the evolution that we can predict based on the model that I gave you. What you can do now is look at a damage model that says that the longer, forget about the equation, the longer the node placed in the region is exposed to a toxic protein, the faster, the more damage it creates. So it's a first order rate damage. It's not just the level that matters, but how long you've been exposed to the toxic agent. And once you do that, you can look and say, well, it's going to create damage at the nodes, and the axon bundle between the two is also going to be damaged. So the weight between two nodes is going to be reduced, reflecting the fact that it's going to lose some of the axon. So you have a reduction of the weight. So it's better seen in picture. What you have, C is the overall average concentration, W is W is the evolution of the weight, and D is the evolution of the damage. So maximum damage is 1, no damage is 0, and W, the weight, is the overall weight, the average weight in the matrix. And so it goes for 1, and then after 30 years decays to 0. So one of the questions I had about maybe a year ago is I said, well, now that you understand transport, maybe one therapy would be to cut part of this connection, and then the disease wouldn't be transported. Yeah, the great idea. Let's try on your brain, not mine. But okay, it's interesting. So the question is, would it change the invasion of the disease if you reduce the transport process? And the answer is no, not at all. So the black line that you see, the dotted black line that you see, is when you don't damage at all, you don't change the network. So there is no evolution of the weight. And the blue one is with evolution of the damage. So despite the fact that the weight in your connection reduces, so that it has actually less transport, there is absolutely no change in the evolution overall in the brain. And we understand why at the mathematical level. Essentially, all you need is a little bit of seed, again, this front propagation that I use for the Fisher equation, to seed the brain everywhere, and even if you cut all the connections, this little seed will just grow to maximum value in time, even if you remove the connection. But we wanted to do more. We wanted to understand cognitive decline. We have a natural evolution of the brain and its weighted connection. And so what you can do now is look at various time points, think about every year, 1, 0 to 30, and you can run a different type of model on this network that simulates the rest state of MRI. So this is like taking a very small snapshot. You have your brain with your connectome, and what you run is oscillator model on each node that represent the normal brain activity when you don't do anything, rest state. You're not thinking about something. You put somebody in an MRI, you say, do not think, which is very hard. Do not think about elephant. Don't think about elephant. Impossible. Elephant's right in your brain. But no, these are rest state fMRI. This is the normal state of your brain when nothing happens. It's a very good baseline, very hard to understand already. And when nothing happens, a lot happens in your brain. You have normal activity, so you have to think of activity as being little oscillator going, sending information left and right, and they are all coupled together, kind of a normal background noise. And when something comes to your attention, in your field of view or hearing, all of a sudden it creates a different type of signal that changes the overall, by the amplitude of this slow background becomes very large and homogenize over the brain, and this is where you take consciousness of something in your environment. So if something comes in my visual field, if it's a ball, I would look at the ball, I would realize it's a ball. There are a lot of process that takes place, and rather than have background noise, there's going to be large amplitude signals, and you can model that. So this is what we do, but on the evolving connectome in order to understand the global change in the brain. So we choose the oscillator in the gamma range, which is an interesting frequency in your brain that's known to be related to this problem of consciousness. And the basic idea is that if the amplitude in the gamma range of the rest state fMRI is too low, then you won't be able to get into that attention mode. And I think, though, there are a few steps there, I think this could be nicely related to the notion of apathy. When the global attention level of a patient goes too slow, the normal rest state, they are unable to realize what's going on around them. The signal that they perceive from the outside is not sufficient to create this global interaction and raise the amplitude to catch attention of something. So you can look at different markers. This goes into area of neural function and neuroscience a little late, so I just want to give you a quick picture. What you see, that's for the entire brain, is that you have a global decline both in the power of T's in the gamma range, but also the amplitude that I told you about. And that you have a different type of staging, different parts of the brain are affected differently. and that goes directly into understanding of cognitive decline of certain type of function with the brain than other ones. So you can be more damaged physically at some place, but some part of the brain, other part of the brain might be not functioning. What you also see is that physical decline always precedes cognitive decline. So there is a natural adaptation mechanism that as this weight decreases, you have more activity in the brain. It tries to make it up by running more the engine, even though the whole thing is starting to fall apart. It's trying to maintain its proper function. So that's the cognitive level remain a certain level where the functional network is already damaged. But at certain time it cannot keep up with it, and then you have an overall global decay, and that's what you observed here. Okay. So there could be, this is an area where we are really excited about because we're starting to understand what's happening to these patients or in the process of this disease. So let me conclude. The highest form of bliss is living with a certain degree of folly. I think you have to be a little bit like that if you want to start applying ideas from mathematics to these problems. These are known to be extremely, every description of it is that these are impossible because they're so complex and all that. And the folly is to try to attempt to extract the simplicity of this process in order to understand general characteristics. So, last slide of conclusion says, in what we are challenged, we try to understand the staging, atrophy, and the biomarker. And so I think now what we have is a platform to try to understand more specific disease, specific condition, and how it's related to cognitive decline. Thank you.
Thank you, Professor Alain. Can anyone here has any questions? We have time for Q&A.
Yes?
Could I ask two quick questions? Firstly, for your last slides, thank you for your talk, it's very interesting. For your last slides regarding the oscillations,